Hi, my name is Mykhaylo Slobodyanyuk, and I'm excited today to present my work titled Direction-Aware Data Fusion Techniques for Multi-Omics Pathway Enrichment Analysis and Biomarker Discovery. To begin, different omics techniques allow us to characterize the various layers of the cell, such as the genome, the epigenome, transcriptome, and proteome, amongst others. All of these different datasets provide distinct yet complementary information about cell function. They don't work individually or independently, but they rather communicate with one another and influence each other in various ways. Thus, it's crucial to jointly analyze these datasets to obtain a holistic understanding of cell biology. ActivePathways, previously developed in the lab, is an integrative pathway enrichment method that uses data fusion to merge signals from multiple omics datasets. First, various upstream analyses, such as differential gene expression, are performed, and p-values are acquired. These significance estimates are well suited for data integration because the upstream processing is expected to take care of technical and experimental confounding effects. To ActivePathways as input, we provide a matrix of gene p-values across multiple omics datasets and a GMT file of functional gene sets, such as gene ontology, biological process terms. A p-value merging method, such as Brown's, is then utilized to fuse the various datasets into a single merged gene list. Afterwards, a ranked hypergeometric test is performed between the functional gene sets from the GMT file and the merged gene list that was acquired from Brown's method to identify enriched pathways. Lastly, the contribution of each individual input dataset is evaluated to see whether the pathways we obtained from this merged gene list could have been identified from each individual dataset alone. Currently, ActivePathways accepts only p-values as input to perform data integration. This is problematic as different datasets may also contain directional information, for example, fold change information from differential expression. In the case of protein and RNA expression, we expect this directionality to be concordant, whilst in the scenario of DNA promoter methylation, we expect more heavily methylated promoters to result in decreased gene expression and consequently decreased levels of protein. Many p-value merging methods exist to integrate various datasets, but Fisher's and Stouffer's methods are the most popular and the most commonly used because they provide the strongest power. So as an example, if you look at the gene CPAD1, we can see that the directionality between datasets is consistent, and both Fisher's and Stouffer's method identify this gene as being significant after merging the p-values. So we obtain a p-value of 0.04 using Fisher's method and 0.02 using Stouffer's method. Another example is the gene called 25A1, which has the same individual p-values for the RNA and protein dataset as CPAD1, but has discordant directionality between the datasets. Inspired by the metal method, which is an extension of Stouffer's method that accounts for directionality, this DI term, published by Willer and others in 2010, we have extended Fisher's method by also providing a directionality term. And as a consequence, we can see that in these direction-aware p-value merging methods, the merged p-value is now no longer considered significant for the gene called 25A1. To summarize, we've incorporated this directional logic into ActivePathways, where in this example we're integrating RNA and protein datasets by prioritizing genes where mRNA is consistent with protein and penalizing genes where mRNA is inconsistent with protein. By doing so, we will remove false positive signals and improve gene and pathway level detection. As our first case study, we integrated HOXA10AS transcriptomic datasets. Previously, Isayev and others in our lab performed experiments with this long non-coding RNA called HOXA10AS. So in a glioma cell line, they knocked down this gene and profiled the entire transcriptome, and then they also overexpressed this gene and profiled the entire transcriptome. Performing two differential expression analyses between wild-type and knockdown and wild-type and overexpressed, they acquired p-values and fold change information for each gene that was profiled in the transcriptome using RNA-seq. And what we expect from the log2 fold change direction between these datasets are opposite signs. So if a specific gene is upregulated in the HOXA10AS knockdown condition, we expect that gene to be downregulated in the overexpression condition. And if we have the same directionality, then we would penalize these genes. 115 genes lose significance when direction is accounted for. If we examine the plot on the left, this line plot, we can see that the x-axis has the original merged p-values when direction is not accounted for, and the y-axis is the modified merged p-value when direction is accounted for. And individual dots are genes, And if they fall along this line, they are not penalized because direction is consistent. And if they fall under this line, they are penalized. And genes that pass this vertical dashed line here are considered significant. So roughly 115 genes of the ones that were originally considered significant are now no longer significant because they were penalized and had inconsistent directionality, whilst 1,024 genes are prioritized. If we further examine the gene ranks of the top 20 genes using the original method, we can see that some genes, like col25A1, go from being the most significant gene to now no longer being considered significant because of directional inconsistency. As a consequence, genes like chRNA1 go up in rank and is considered the top most significant gene. Some other genes, like col9A3, only shift in rank slightly and are only slightly penalized, seen here. This change in gene rank, as well as in the number of significant genes that we identify, influences the pathway level findings. So using the original method, we can see that some of the pathways we identify include ossification, NCAM signaling, transforming growth factor beta production, protein catabolism, muscle tissue development, mitotic cell cycle, collagen formation. But when we account for direction, some pathways are lost. So entire themes like NCAM signaling are lost, collagen formation, mitotic cell cycle, whilst other themes are fully retained, like protein catabolism, muscle tissue development. Not a single pathway within this theme is lost. Some pathways are also gained using the direction-aware method, such as integrin cell surface interactions and positive regulation of protein ubiquitination, amongst others. We performed another case study on the renal cell carcinoma cohort from Clark and others, deposited in CPTAC. So we performed medium dichotomized survival analysis using both RNA and protein data separately. Afterwards, p-values and log2 hazard ratios were acquired, serving as a proxy for directionality between these datasets. And we expect RNA and protein to have the same direction. Those genes with opposite log2 hazard ratios are penalized. On the line plot on the left, we can see that about 88 genes are penalized and lose significance because they have inconsistent directionality, and 764 genes are prioritized. If you look at the top most significant gene, SHC1 or SHC transforming protein 1, we can see that if we look at the RNA expression Kaplan-Meier plots, high expression of this gene produces a high-risk group. For protein, a high expression of this protein also produces a high-risk group. So there's this consistency between these two data sets. If you look at the gene MTF-MT, this gene is a mitochondrial methionine tRNA formyltransferase, and it has inconsistent direction between RNA and protein data. So in this Kaplan-Meier plot, low expression of this gene produces a high-risk group, whilst high expression of this protein produces a high-risk group as well. And for this reason, this gene was penalized. If we look at the pathway level signal using the original method, we can see that certain pathways are enriched that are involved in renal cell carcinoma patient outcome. So these pathways involve protein translation, viral transcription, protein localization to mitochondrion, neutrophil and leukocyte-mediated immunity, cellular detoxification, amongst others. If we account for directionality, certain pathways are penalized, so the entire theme of cellular detoxification is lost. Interleukin-12 signaling is lost, amongst other pathways. Some pathways actually gain further support. Pathways like neutrophil and leukocyte-mediated immunity don't lose a single pathway, and they gain an extra pathway when direction is accounted for. The same goes for cellular protein complex disassembly, amongst others. So to summarize, we provide a flexible framework to integrate data by additionally defining the direction of interaction of genes in different data sets. This tool is applied to a HOXA10AS case study to integrate differentially expressed genes and prioritize key biomarkers and pathways implicated in HOXA10AS function. This tool is also applied to the renal cell carcinoma case study to integrate prognostic genes from RNA and protein datasets and prioritize key biomarkers and pathways implicated in patient outcome. The modified ActivePathways method can be applied to a variety of different datasets and analyses where p-values and directionality information can be acquired. And with this, I'd like to acknowledge the Raman Lab, my committee members, OICR, and Medical Biophysics for funding. Thank you so much for listening to my project.